Thank you. Thank you. Yes, we are a Canadian slash French company because our team is split between Europe and North America. Thanks, everyone. It's great to have the opportunity to participate in the forum today. It's great to be back here. I'm going to speak less about the company and more about just about highlights of what work we've been doing recently. There's a little bit more time for discussion. I'm excited to share the progress that we're making in our neuro recovery therapy development. So let me just make sure if I'm sharing the screen here, we can click ahead. Okay, so for this audience, I don't think we need to spend a lot of time talking about the need for new therapies, but briefly, 1 in 4 of us is going to suffer a stroke in their lifetime now. And the vast majority of folks are going to still be left with some type of life-altering disability. Those are surviving are not necessarily those that are recovering well. And 100 million people worldwide are living with after stroke or traumatic brain injury. So the good news is you're more likely to survive a stroke than ever before, but the bad news is that you're more likely to be faced with these lifelong problems, right? So, and yet we know that the brain has this intrinsic ability to rewire itself and reorganize after injury. It's frustratingly difficult to solve for, but we need to be reminded how dramatically fast and effective this can be, right? So, and this is as a source of where some of our development comes from in Panaxium. So for example, our chief medical officer, Dr. DJ Cook, DJ's the head of neurosurgery at Queen's University and leads the translational stroke recovery lab, where we do a lot of our primate work in Canada. And what he's doing in his practice is coaxing a lot of remapping, reorganization intentionally as part of tumor resection procedures, right? So this is to move function that will be lost from the resection. And you can observe that within timeframes of weeks, entire functional areas can be moved to other areas of the cortex in response to intentional interventions, so that patient can completely avoid any deficit in many situations when an infiltrating tumor is fully removed, even if that originally infiltrated into functional areas. And we can see that function moving in imagery. It can happen incredibly fast. Frustratingly, we got to figure out a way to do this for people who come in not with an intentional injury, but what do you do when someone comes in presents with a stroke, with a traumatic brain injury, right? And this is not a new concept. So when the primary function is lost, in the case of M1 and the motor cortex, the brain tries to remap to elsewhere, right? And we're just promoting and expanding that remapping into those secondary areas, right? And if you do that, if you stimulate, this is a mechanism that we're using in a lot of different approaches, you can see growth in those other alternative areas. This is actually not the best alternative area, so don't judge me on where that's located on the brain. And so our solution is focused again on direct cortical stimulation, but we do this with very high precision, very high, relative resolution and stimulation control directed at the cortex, right? So our solution is a temporary implant that's really ultralight and thin and conforms to the surface of the cortex very well. So as you can see here in this example, you can see it kind of folds over into the sulci. It wicks well to the surface, makes a very good biomechanical interface. But what really makes it special is that we focus a lot on components at the surface that are made by exploiting special materials like ion conducting polymers. In terms of high resolution, if you're looking at this image, don't be focused on these relatively large 1 millimeter reference electrodes. When I say high resolution, we're talking about ones targeted to the resolution of cortical columns, things that are more like 100 microns. We do things that are even smaller for other devices, although not necessary for this purpose. And so we're using this activity-dependent plasticity, right? Stimulation reinforced with some type of paired therapy, task therapy, and which is the basis of many, many different types of approaches. The big difference is that we're making use of this high-resolution stimulation over relatively large functional areas. And the one takeaway from today is that the resolution does matter. So we're not trying to activate or potentiate everything at once. What we're doing is mapping these points that are prime spots for reorganization, targeting them and focusing our interactions on those with our device. And this is the key difference here is that spatial and temporal matching of the stimulation to the response of the areas of interest makes a big difference. So these are representative pictures of human motions. This is actual data from a primate. And what you'll see is that the same motions are going to respond, map differently in the same areas. These things are intertwined, but they do change spatially and they do change temporally. So the selectivity in those dimensions is very important. So how do we do this with our device? What makes our device different? The key thing here is that we rely on some interesting materials in our devices. Our team is really kind of world leaders in the field of conductive polymers for neural interfaces, and not just using them as an alternative electrode material, but really is being able to build components out of those materials on the scale of the device. And so what makes this difference is that they allow the transport and response using kind of these ions, natural electrical signals from cells. And that effectively means that you can do this with less power and you prevent side effects that you would expect from conventional electrodes like electrochemical damage or thermal damage. And that means we can kind of achieve motor thresholds at much, much lower currents. So this breaks the kind of convention. And also we have the ability to deliver neurotransmitters or small molecules with these devices. It kind of breaks that conventional thinking of what neurostimulation is, because we can use this to deliver things like GABA or acetylcholine, small drug molecules as well. So there's a potential for combination therapies, although not necessary for this initial mechanism of action. The other thing about this, and there's a lot of effort we have a team in the company that's working on this, dedicated to focusing on imaging, because it's not just about the hardware, right? So if we want to achieve a good therapy, we need to understand the injury at a similar scale. And a critical component of this is the development of some analytical models that we can apply to each patient to understand how their personal injury is disrupting their functional networks, and use that to predict how it could recover, whether it's with our therapy or not, and then plan a corresponding intervention. And there's just, this just doesn't exist today. An example here of this patient, you can see the lesions marked in green, where we've scanned and analyzed, and you can see then how it, analyzes how it affects the entire network, and then allows you to actually predict recovery and also tell us where's the best place to go stimulate, because it's not always in the same spot. So the idea here, 3 steps. There's a neuroimaging assessment that's critical to the way our assessment and planning. There is the delivery of our stimulation implant. And then there is a follow-up of this AI monitored therapy, right? This AI monitored, the device has the ability to actually do recording as bidirectional. So that allows for the potential for closed loop reinforcement outside of paired physical therapy. Although not the immediate regulatory hurdle we're planning to tackle out of the gate. So, this is not a new mechanism. There's variations in terms of what we talk about in terms of this kind of spatial and temporal precision. But we've seen this type of direct cortical stimulation before. We're all familiar with the North Star Everest trials from years ago, many other kind of clinical research studies since. There are 3, and forgive me, don't shoot me, because I know this is nuanced, so I'm not trying to oversimplify it. But generally speaking, if you can evoke a motor threshold, then generally there's good prospects to help with recovery. And there's been limitations that we've helped overcome. So, and again, this all kind of comes back to like being able to have a higher resolution device where we can evoke precision responses. And so one is reaching the motor threshold. So we're able to do that with lower currents and be able to target that with a resolution so we can achieve that more readily. We can achieve the motor threshold for a movement you want to assist to help in recovery without causing the side effect tremors of other movements, because we're able to be selective about what we're evoking and what we're not. And then we're also doing this, as I described, proprietary AI-assisted imaging analysis in order to just pick and predict recovery in patients and pick the best candidates for this therapy, and actually then allow us to have a broader number of patients that we can go and target. Okay. So I won't dwell on all this. This is just one kind of example. This is work in primates, but just highlighted one example here is that in this case, again, these are kind of the human equivalents, but the elbow could be motor threshold is only 0.2 milliamps, where the wrist was 0.8 in this particular example. And you can see these things spatially, they're very close together. So I'm just focusing in on, as an example area where we did some stimulation over here in PMD, target area for remapping. And what you would normally do is that if you're trying to help that wrist motion and you go to reach that close to the motor threshold and set some type of like stimulation just below that, then you're going to be super threshold in the elbow several times over. And what happens is you're essentially going to cause tremor in one motion without before you can achieve the other. So but we can pick and choose these separately. And it's not so simple just saying one spot. It's combination of areas. So you have to like pick and choose combinations of electrodes. It's an oversimplification to say that you can pick one dot right beside another. But generally speaking, we have the ability to map these areas for the different responses and correspond our stimulation to each one. Now, I'm happy to go through all of that data. If someone wants to come and talk to me, I'm happy to go through it in very detail. I just want to share a couple other updates about what we're doing as a company. More recently, we've signed a co-development agreement with a partner company, Synaptive Medical, who's a real innovator in neurosurgical navigation and guidance systems. And they also have their own MRI systems, the Every systems, which are really fantastic results. So I'm just putting a plug in for Synaptive, showing kind of deep brain structures better than you would see in higher field magnets. So if you're into DBS therapies, I suggest you go check out what they're doing. And they approached us to integrate our devices, our platform for electrophysiology recordings in their systems, which is tremendously exciting because this allows us the ability to do high resolution mapping, which is a very important element of the overall therapy, over large areas of co-registered to other imaging modalities. And so this agreement gives us a great opportunity to pipeline of new KOLs. It's getting us feedback sooner and really a platform for generating a tremendous amount of great data. Some of that data is we're going to be getting out of a clinical trial that's beginning later this year in Paris. We have a 50-patient trial using our devices, again, interoperatively. It'll be the first in human use of our devices for that purpose. So we're just going through that kind of like the final steps of that. And it'll be a pretty rapid study that we're looking forward to. And then where we are generally is we have preclinical primate stroke recovery studies underway right now. So looking forward to seeing the results of those over the next few months. We're collecting in parallel more and more neuroimaging data from a number of stroke patients at 4 research hospitals, 3 research centers, all in Canada. And then we have this, as I said, this 50-patient trial for interoperative monitoring, which is going to allow us to get a whole bunch of great kind of human mapping data from it. So that's where we are today. We are always looking for partners who want to help us. We know we can't do this all on our own, always looking for new investors as well. That doesn't stop. And looking forward to pushing this into trials sometime in the relatively short horizons. Thanks.
Thank you. Thank you very much. Brad, a quick question for you. Can you, first of all, stop share? Because Sandra's going to be up next.
Okay.
And Sandra, you can go ahead and... Is this working? Okay. So, interesting presentation. I'm not sure, were you here for Helen's keynote
Yes.
talk? So one of the things she mentioned, which I thought was really, relevant, is speaking from a clinician's point of view. She has a different perspective than what the hardware, developer will have, and sometimes we tend to get bogged down in the hardware details and not enough in how the patient is responding. And so how would that impact Panaxium in terms of the evolution of your product, in terms of what changes you'd be looking for, or what feedback you would want from clinicians and patients?
Yeah. So a couple of things in her talk, and one thing that resonated very strongly was the need for imaging, but also the challenge of getting that imaging, because if it's not part of conventional workflows, how do you get that? Because most patients today do not get MRIs after they've had a stroke, let alone at the right time, so that you can assess what candidacy they have for a therapy. So we see some changes in that, but I think that the changes are actually in that area are actually going to come from greater use of MRI on presenting patients in the ER, because there's studies now that are showing that the MR is much, much better than the CT. And I think we kind of have to piggyback on those trends, because convincing people to do a lot more imaging is hard. So we're sensitive to that. The other one we're sensitive to is that everything's paired with therapy. Paired some physiotherapy. So you have to think about how this would ever interact with the therapy clinic. And ultimately, we got to find a way. The real kind of like holy grail is to get this out of the clinic and be able to do it at home. And I think that's where the closed feedback is very exciting. And it's one of the things that's attractive about cortical stimulation and recording, is that the device allows you to kind of do both. And so you can get those biomarkers from the device that's present. Again, that's a little more challenging, that our first version of this as a product is not going to have that closed feedback, but all the components to be able to build that exist today.
Got it. Okay. Do we have any questions? Jojo, does anyone on Zoom have a question? No, but we do have a question in the room.
Thanks. Fascinating presentation. Love the technology. Tell us about resolution as much as you can, in terms of with what resolution can you stimulate versus sense? Can you sense individual cells? Can you stimulate individual cells?
So we can. So for example, in our intraoperative work, the electrodes, it's a very special configuration to collect certain biomarkers from high frequency oscillations. Those electrodes are only going to be, or those contacts, we're hesitant to call them conventional electrodes, are only 30 microns. We can make them smaller, but you're talking about ones that are the size of neuron bodies, right? So we can kind of make them as small as we want. Practically speaking, the limiting factor is how do you combine that resolution with the ability to evoke responses deeper within the cortex? You don't need it. So we kind of focus on the hundreds of microns. So we can kind of do it whatever we want, but we need to have the electronics to be able to access those and to stimulate them properly, which is there's real limitations right now. We think that in those hundreds of microns of dimensions, at that point, it's better to get more coverage and be able to focus on areas outside of just M1 and PMD and PMV and parietal area 5, and be able to get that coverage sufficiently. And so that's kind of the sweet spot that we're addressing.
Okay. Thank you very much, Brad.
Okay, great. Thanks.